2 to 3 temperatures over Celsius, cooler than the core body temperature to make active sperms. And that is done with the premaster muscle. Sometimes it contracts and sometimes it relaxes. Once contracted, it brings the testis very close to the pelvic cavity, and that is for warmer temperature. And when it relaxes, it pushes the testicle or testis away from the pelvic cavity, and that is for a cooler temperature. I have at least 5 questions back and forth and back next week. The same exact idea. Premaster acting as thermostat, skeletal muscle. It can contract, it can relax. Upon contraction, it brings testis closer for warm, right? Warmer. And upon relaxation, it sends them away, and that is for cooler. At least 5 questions revolving around the same exact piece of information from slide 7 on the lecture exam next week on Monday. The sketch here, I add that just to show the symptoms, so it's not of any importance, just to show you, so this is just extra. Visual aid. I was just looking at the term septum, which is by division, that divides the scrotum, as you can see, into 2 sacs, and each one is holding inside it 1 testis, 1 gland for making sperms and making also many hormones. Slide 9, please, and testis is my title. Please highlight for me everything, and also highlight the sketch. What does pair mean? Paired. 2. What is the equivalent for testis in female? Ovary, right. And what is the equivalent for sperm in female? Egg. What do they both have in common? Egg and sperm. Gametes, which means how many chromosomes in total we should have? 1, 2, 3 all, right? So remember we have 46, right? You should have 46, with the exception of egg and sperm. They both should have only 23. It made them a haploid, wasn't that right? Haploid, so 1N or haploid, it only carries 23 total number of chromosomes. Next is a third over gland in the scrotum. If you look inside the testis, if you look inside, you see those coiled tubes, we name them seminiferous tubules. So each contains seminiferous tubules. Seminiferous means sperm carrying. So we have sperm carrying tubules. And sperm production happens here by a process named spermatogenesis. What is genesis as a suffix? Genesis? To create, to make, right? To produce. So this is the process of making sperms. It happens inside the tubes, which we name seminiferous tubules by the process, and that is spermatogenesis. But then if you continue coming out of it or sitting right on top, we have our next structure, and that is epididymis. Epididymis is made up of 3 sections. Please notice head, body, and tail. So we start with head. This is the body, and then we end up with tail. And if you continue, it turns into a tube, and that tube is named vas deferens, right? Epididymis. Epi means above. Didymis means testis. Above testis, or epi, which is the same structure we had for the lab exam, which is right here, slide 6. Epididymis, it sits right on top. So epi means above, didymis means testis. So sperms are made, we move them and store them inside epididymis. Now my fine print indicated that we have head, we have body, and we have also tail, right? And then it continues as the long tube, and that is vas or vas deferens or ductus. We're going to take a look at those in a second. So that is for testis. Let's take a look at the process of making sperms, which is termed, again, spermatogenesis. Before that, let's take a look at the types of cells we have inside testis. I have 3 different types of cells. I have spermatogenic cells, I have Sertoli cells, and I have leavage, or sometimes named interstitial cells. Now let me start with number 3 and then walk up. So we're going to start with leavage or interstitial. Those are found in spaces between the seminiferous tubules. And the most important function for leavage is making the hormone testosterone. Now, why do we sometimes name them interstitial? Because this is my micrograph of the seminiferous tubules. So I have tubes in the testis, and look at the leavage. They're always found in spaces in between. See how they are hidden? Hidden in the space in between, which means interstitial in between. So sometimes we name them interstitial. I do prefer leadage after France, leadage from Germany. So leadage or sometimes interstitial. It is where the hormone testosterone is coming from. My second type is Sertoli, and it has a long test memorization list of functions, including nourishing the spermatocytes, spermatid and sperm. What does nourishing mean? Bringing in, like feeding, right? Feeding, nutrition, exactly. Number 2, phagocytizing excess spermatid cytoplasm. What is phagocytizing? Absorbing or eating out extra cytoplasm. Number 3, controlling the movement of spermatogenic cells. Number 4, releasing sperms into the lumen and producing the fluid that will help in sperm transportation. And then lastly, I have making the hormone. This is the most important, the hormone inhibin, and that hormone is looking after both testosterone and mostly important, looking after follicle stimulating hormone. So we have a long list of functions for Sertoli cells. So where about did I make my testosterone? It was made in interstitial or leadage cells. And where about I made my hormone inhibin? That was made by Sertoli cells, right? The first type is my spermatogenic cells, and this is where sperms are coming from by the process named spermatogenesis. You don't have to really worry about it. Because I made a flow chart to show you how sperms are made, and this is our next topic, which is my title, spermatogenesis. So please highlight everything. You need to skip on this part here, because I want to simplify for you and make it quite easy. We're going to skip on what I outlined and highlight everything else, please. How many days does it take to complete a cycle? Between 65 and 75 cycles on top. It takes between 65 and 75 days to make active sperms, roughly over 2 months. Before I go into the conclusion, which I made in green and in blue, let's take a look at my flow chart that summarizes what I have here. So this is not deleted, this is just summarized in a much easier flow chart to digest. So this is the process of making sperms that takes between 65 and 75 days to be completed. And then this is how it begins. It begins with, I have my cell type, as you can see, transitioning, moving down. We start as spermatogonia, stem cell. Now please pay attention to my fine print. So is that diploid or is it haploid? Diploid, which means 46 chromosomes. So this is my fine print to focus on as well when you take it home and study it. From spermatogonia, we make primary spermatocytes. Are those dip or hap? Dip. Excellent. From primary, we made our secondary spermatocytes, and those are now haploid. From this, you make spermatid, of course, haploid. From that, you make your mature sperm, still haploid, and then you end up with making the freely swimming or freely moving, free willy, isn't it? Free willy, or freely moving sperm, and that is also one end. My fine print also to focus on are the stages in between. So mitosis, meiosis 1, meiosis 2, spermiogenesis. What is spermiogenesis? It is the way you change the spermatid into mature. What is spermiogenesis? Well, I can give you my definition. It is on slide 11. My spermiogenesis explained. It is the development of spermatid into a sperm. In that case, a spherical spermatid will be changed into something more elongated. We have mitochondria. We have our acrosome. We also have our flagella. So this is formed. Mitochondria is formed, and the acrosome is also formed, and it does change from being what does spherical mean? Brown, which is like just like this here. I'm going to jump in for a second, so you can also take a chance of the highlights. Just move it out of the way. My head, big piece, and tail from our lab exam right here. Please notice how the spermatid is spherical. This is how it begins, right? Spherical, and then as you go through the stages, it becomes more elongated. The process by changing spherical to elongated, also making sure you have the acrosome and the mitochondria and the tail, That is called spermiogenesis. And then from mature, we have to make it freely swimming, and that is spermiation. So what is spermiation? Back to slide 11. It is basically the releasing of that sperm from its connection to, it's just to set it free. To set free those sperms, we name it spermiation. Can slow down and rewind one more time. I think I have 3 different types of cells in the testis. Is that right? Yes. What type of cells we call nourishing, transporting, making the hormone inhibin? Serotonin. Which one is responsible for making the most important hormone in male testosterone? Interstitial or leadage cells. And where do sperms come from? Well, they come from the first time, isn't it? Spermatogenic cells are good for making and producing sperms. How long does it take to complete one cycle for making sperms? 65 up to 75 days, and this is how it works. We start as spermatogonia stem cells. By mitosis, we change into primary spermatocytes. By meiosis 1, we change into secondary spermatocytes. By meiosis 2, we change into spermatid. And by spermiogenesis, again, what was spermiogenesis from slide 11? It is, as you can see, how the spherical turns more elongated. Mitochondria is now available. Acrosome is formed, and we also just made the tail or the flagellum. And then we have to set those free, right? To set free is the process of spermiation. We have to release them and separate those from their connection to serotonin cell, and that was my last step. From mature to free, and the process is named. Okay. And my fine print again, we go from 2N, and then 1, 1, 1, 1, all the way. So only the 2 guys, the first 2 guys are diploid, the rest are all haploid all the way down. Sounds cool? And that is very easy compared to the complicated way to explain it. With my information, I haven't that highlighted or outlined part, as you can see. The first part of slide is 11. Very good. Next, we're going to continue with sperm then. We're not done with sperm yet. And let's take a look at slide 14, please. And let's look at 15 first and go back to 14. Please take your time and highlight. This is a closer look at sperm. A much closer look at sperm. From sperm, we jump to hormones next. We know, of course, from our lab exam, it comes with a head and it comes with a tail, right? But that tail is made up of 2 substructures, a little neck, and most important, a mid or middle piece. What do you see within the head? I see nucleus. It has in it 23 chromosomes, as we know. But most important, I can see a little sac named acrosome. Now, if I open that sac, I can release very strong hydrolytic enzymes needed for the penetration of the egg and for fertilization to take place. Please allow me to slow down, rewind, and read out the same information highlighted one more time on slide 14, please. Each day, about 400 million sperms are completing the cycle of spermatogenesis. As a matter of fact, in 1 milliliter of semen, we have between 50 and 150 million sperms. And then we have 2, as you can see, 2 parts of the sperm. We had the head, and we had also the tail. The head has in it a nucleus, and it has in it also an acrosome. But how many chromosomes we had in total within the nucleus? 23 chromosomes, which made it a haploid or 1N. And what makes an acrosome so important? It is just a sac or vesicle that contains inside it the what is an oocyte? An egg from lab, oocyte. My oocyte penetrating very strong digestive and hydrolytic enzymes. You can see them hidden and stored inside the acrosome. The tail has in 2 parts. I have neck and middle piece. The middle piece contains mitochondria. What is my need for mitochondria? For movement, right? ATP, for the sperm to swim and find its way. My neck contains centrioles that makes microtubules, and that is for the making of the flagellum or we know from part one that cilia and flagella both are made up of microtubule, not microfilament, not intermediate filament. So if you go back to part one, we know that cilia,
We're done with it.
cilia or cilia, flagella or flagella, plural, both have in common the fact that both are made up of microtubule. So in the neck we have microtubule to start making this long tail, and that tail is no more than a flagellum. And then we have mitochondria, plenty of mitochondria, as you can see hidden inside the middle or middle piece. Once ejaculated, a sperm does not survive more than 48 hours in the female part. A long time, isn't it? Some can go up to 5 days. Some can survive up to 5 days after ejaculation, and we call them, what do you think we call them? Super sperms, Diane. This is not a joke, this is information. So some can survive up to 5 days, and we name them super sperms, but for the most part, up to 48 hours, we cannot survive more than 48 after ejaculation takes place. Next, we have our hormones, and we have totally 4 hormones to look at. I have them in blue and pre-underlined. Just skipping some numbers first. So the first hormone to look at is LH, number 1. Secondly, I have FSH, number 2. Third, I have inhibin, number 3. So where's number 4? Number 4 is on slide 17. In the end, the most important, and number 4 I have for, as you can see, TASA, TOSIN, T1, the longest list of functions. Let's start with the first one, and I have luteinizing hormone. The function is to stimulate Leydig cells to make, what's it Leydig that make this testosterone for me? Is that right? Leydig? Okay. Can I go back and confirm with you? So inside the testis, what type of cell made TASA, testosterone? Leydig made TASA, so Leydig confirmed. So it is basically working on LH, or luteinizing is working on Leydig for making more and more of the hormone testosterone. Secondly, I have follicle stimulating. Secondly, yes, follicle stimulating hormone. It does act indirectly on the process of making sperms or spermatogenesis. How does it work? Let's find out. FSH and testosterone both act on Sertoli cells to make the protein named antigen-binding protein. Once that is made in high concentration, it binds to testosterone, keeping it the concentration extremely high. Once testosterone concentration is brought up and becomes really more abundant, it will stimulate more and more of making sperms in the process we name spermatogenesis. Next, we talked about Sertoli, we talked about making inhibin, we talked about looking after the hormone FSH. But the most important hormone of all of those, of course, is number 4, and that is the most abundant, the most popular hormone in the male system is testosterone, with the longest list of functions, including, this is just a matter of highlighting these and reading out, nothing more. A development of male sexual properties. At puberty, it brings about the development of male sex organs and development of male secondary sexual properties such as voice deepening, hair distribution, an increase in bone mass, also an increase in muscle mass. Secondly, a development of the sexual functions. It can contribute to male sexual behavior, making more sperms by spermatogenesis, and also the sexual drive. And then lastly, the stimulation of anabolism. What was anabolism? Making, right? Building up or making, as a matter of fact, it does stimulate making more proteins, and that is for a heavier muscle mass and more bone mass. So what hormone is looking after FSH? Looking after FSH? I think inhibit, wasn't it? Which one was looking after FSH? Inhibit, when I say inhibits means it controls. Controls how much you need of FSH. So which one is looking after FSH? Inhibin. Which one is good for bone mass and muscle mass and sexual drive? That's testosterone, the most abundant. Which one is good for making more sperms? FSH, isn't it? So FSH makes more sperm by the process spermatogenesis, and which one is good for making more of the hormone testosterone itself? Utilizing hormones. So those are the four hormones in the male system. Each one has a specific function, of course, in the male reproductive system. Very good. Please jump with me to slide 18. 18 and 19 both are fully highlighted, and now we have the system of ducts. And we have very glad to look at system of ducts in the male system. Ducts of testis, the pressure made by the fluid produced by circulatory cell will push sperms along seminiferous tubules and into the epididymis. And then we have totally 4 ducts, and those are pre-numbered. I have them also pre-underlined in blue. I have epididymis. We will take a look at epididymis in a minute. We have vas deferens. We have also on the next slide ejaculation duct, and we have the last one, and that is urethra. Now this part here, please highlight first with me, and let's move it out of the way first. Again, what is the ascent? Moving up, right? Moving up from the scrotum. We have this is not a duct, this is just something, I don't know, they have to on the same slide, but it just happened to be on the same slide by mistake. It's not belong here, this is a different structure on its own. But ascending from the scrotum, we have a spermatic cord. If you look inside it, you can see vas deferens, you have artery, you have vein, you have a nerve, you have a lymphatic vessel, but most important, you also have a pre-master muscle. Please stay with me and let's confirm it again. So that was my sketch from slide 8. Ascending from the scrotum, we highlighted a spermatic cord. Again, you make an incision and peel it, look inside it. You have inside vas deferens, you have a nerve, you have a vein, an artery, a lymphatic, not shown, but a lymphatic vessel. But most important, we have the thermostat, and that is the skeletal muscle named pre-master. So this is all included inside that tube that comes up from the scrotum, and that tube is named spermatic cord. So this is just to move it out of the way and focus on ducts. The ducts we have here are the following. We have the first one, and that is epididymis. So the first one is epididymis. Again, what does it mean, epi? Above, didymis, testis. It is right on top of testis, and it is made out of tightly coiled ducts. As a matter of fact, it is up to 20 inches in length. Wow. So epididymis, if you switch it, it can come up to 6 meters or 20 feet in length. 20 feet in length, sorry, 20 feet in length, or 6 meters. It is a site for sperm maturation because you move them there and they can acquire, what does acquire mean? Skills like learn how to become more mobile or in motion, also ability to fertilize. You can store sperms in epididymis for several months, between 1 and 3 months only. You can store them between 1 and 3 months, and then what happens if they still sit there? You have to get rid of those sperms, so they end up being worn out. And then once they're worn out, you have to remove them. And the removal is happening by stereocilia. The name is cilia, isn't it? But it is not a cilia. It is actually a microvilli. This is my epididymis. Just looking inside it. Can you see the lining of it? Of epididymis, the lining. This is my stereocilia. So even though the name has cilia in it, it is a variant of microvilli, and the function of that microvilli is to target those dead, worn out sperms and suck them in, absorbing them to get rid of those unwanted extra load, of course, extra load on the epididymis. We have to empty it before we empty new number of active and healthy sperms in this structure. So this is good for the maturation and the movement of the sperm. Good for storing sperms between 1 and 3 months only. If ejaculation is not happening, they end up dying, and then how can we get rid of those sperms? By absorbing them by the line, and that is microvilli to reabsorb degenerated, worn out sperms. And it continues as vas deferens. Is that right? Rather than going back and forth in my slide, I have the same sketch, which is easy for me. So that was the testis in which sperms are made, right? We leave them and we store them up to 1 and 3 months, right? In epididymis, and then this one continues as vas, right? So it does continue as you can see. So the next structure for sperms to move in is vas or ductus deferens. Vas means vessel. The term vas means vessel, and ductus means duct. Duct, vessel. Deferens means to carry away. Means carry. Topic, we don't have any print out, so this is my carrying away vessel. Carrying away duct. And this is my translation. So again, duct is ductus, vessel is vas. The term difference in Latin means to carry away. Okay, so this is my carrying away vessel or my carrying away duct. It conveys, which means it moves. It moves sperms during sexual arousal via peristaltic. Is that something we learned before? Peristaltic movement, peristalsis, which means if you take out this structure, if I can take out the vas deferens and look at it in a cross section, don't expect any of these smooth muscles arranged in two sections. One is circular and one is, it has to be, just like your esophagus, the same anatomy. So whenever I hear peristalsis, it has to have two layers of muscles for contraction, right? This is on the exam, I promise, next week. This is on the exam. So how do sperms move from epididymis all the way to the ejaculatory duct? Does it move by gravity? No, this is moving up, isn't it? So there's no gravity. Does it move by skeletal muscle contraction? Okay, a skeletal muscle in that picture? No. Or does it move by the smooth muscle contraction in the wall of the vas deferens? Smooth muscle contraction, like milking contraction, and vas deferens, smooth muscle milking contractions, and vas deferens is good for moving and taking sperms from epididymis all the way to the ejaculation or ejaculatory duct. Peristalsis equals smooth muscle contractions. It has to be smooth muscles. And then it can also store sperms, but only for a month, right? This is 1 to 3. This is only for one simple month, nothing more than 1 month. Next, I have ejaculatory duct, and then we're going to end up with something we learned in details in the kidney, and that is urethra. So please stay with me for a second. We make sperms, I think, in the testis, right? And where do we store them? In epididymis. For how long? 1 up to 3. And then when ejaculation takes place, you empty epididymis. Sperms are milked into vas deferens, and we are right here, folks, at that end. The next structure is this tube, and that is ejaculatory duct. How is that made? If you remember from our lab exam, we have a gland named seminal vesicle, wasn't it? Right next door is vas deferens. These two guys united to make one duct, and that is ejaculatory, wasn't it? Duct. And we think this one ends in urethra. Isn't that the beginning of urethra? So this here, this is urethra, isn't it? All the way down. So can I say that this one terminates at urethra? Yes, one more time. This guy is vas deferens, a long tube. This guy is a gland named seminal vesicle. When seminal vesicle meets with vas deferens, when these two guys unite, they make one duct, and that is ejaculation duct. That does end in, of course, urethra. Please stay with me for a second then, so let's confirm it. It is formed by the union of seminal vesicle and vas, is that right? Vas deferens unites with seminal vesicle to make the ejaculatory duct? Yes. It also does terminate in prostatic, is that right? Does it end in prostatic urethra? Yes, it did. Function is ejecting sperms and seminal vesicle secretion just before releasing of semen into urethra. Lastly, I have urethra, a shared terminal duct of both reproductive system and chapter 26, urinary system. It is made up of how many sections? 3. Prostatic, membranous, and then penile, sometimes named spongy urethra, and it ends at the external urethral orifice. Does that bring numbers from last week, the last exam? I think so. So here comes my ejaculatory duct. It terminates at urethra, but what part of urethra it ends at? Prostatic, right? So please stay with me. Prostatic urethra. This tiny little area is membranous urethra. The longest section is named penile or spongy, right? Spongy or penile, it ends in the external urethral orifice. So the exit of urine and semen is that opening, and that is an external urethral orifice. Perfect. We don't have to highlight it if you don't want to. This is never on the exam, it's very complicated, but sometimes it's easy to understand from a different view, right? So let's just stick with one single view. So if you adjust your brain and look at it from a different view to understand all the stuff we talked about all day. So this is not for the exam, just FYI, so please stay with me and let's take our tour from A to Z. Where sperms are made is named testes, right? We move them and we have them stored between 1 and 3 months inside this little structure named epididymis. Please stay with me. Ejaculation is taking place. Sperms are moving into this tube, and that is vas deferens. So we're moving down. Vas deferens. This is the end of it, isn't it? Right next door we have seminal vesicle, the gland. When these two guys united, what did they make? They made ejaculatory duct. It will empty sperms and semen into urethra, which is the first part, and that is prostatic urethra. This is membranous urethra. The longest section is named penile or sponge urethra, ending in the external urethral orifice. Also, since I am on this here, can you see the two tissues of penis, the cavernosum and spongiosum? The inside wasn't the spongiosum to the inside. The outside it was cavernosum. So we're gonna learn in a second about the structure named glans venus, which is that little swelling at the end. Glans venus. Based on my sketch, and keep this piece in the back of your mind for a minute or two. Based on my sketch is glans, this structure, the glans venus, is that the enlargement of the sponge or the cover? Does it come from spongiosum or cavernosum? I love you. So please stay with me. This is the sponge, isn't it? Sponge, it's moving down all the way, and then at the end, it does expand, doesn't it? So in the back of my mind, yes, it is the sponge that does expand. Once it swells and expands, it makes our glans penis, which I will talk about in a couple of minutes. Are we good? Next is, we're almost done. We have our glands, and slide 21, please. We have 3 important glands that makes most of the liquid portion of semen, including seminal vesicle, prostate, and vulvourethra, also known as Cooper's. After the surgeon from England, the UK, Mr. Cooper. Cooper's or vulvourethral gland. Percentage is important for the lecture exam next week. So what percentage of semen comes from seminal vesicle? 60%. Please emphasize on it. And what percentage comes from prostate? 25%. Please emphasize on it. And what percentage comes from Cooper's or vulvourethra? Only 15%. So I have 60, 25, and I have 15%. Let me start with the first guy, and that is seminal vesicle, which makes up to 60% of the fluid that comes with semen. It makes an alkaline, viscous. What does viscous mean? Thick fluid containing fructose. What is fructose? ATP. Why do I need fructose? For making energy, ATP. If you don't have this, please add this to your food now. So I have in it fructose, which is a sugar. You can burn it and make energy. So ATP comes from burning fructose. It has in it chemicals named prostaglandins. What are those good for? Those are good for sperm viability. What does viability mean? To stay alive, right? Keep them alive, keeping sperms alive. Viable means living or alive. So again, I have chemicals named prostaglandins. Those are for keeping sperms alive. Or you would say, for sperm viability, the same thing. I am slowing down because I want to make sure that you guys match exactly, because sometimes I edit and I add stuff, so please match your PowerPoint with what I have on the screen. And then I have some clotting proteins, something like seminogillin, that will cause semen to clot immediately after ejaculation takes place. 60% comes from seminal vesicles, is that right? Yes. It contains something for making energy, and that is the sugar fructose. It has something to keep sperms viable and alive, and thus this is a family of proteins named prostaglandins. It has a chemical that can arrest, when I say clot means to stop, right? Arrest sperms right after ejaculation, something like the protein seminogillin. This is basically what I have in seminal vesicle secretion. Next, I have prostate, and it counts up to 25% of the volume of fluid of semen. It makes a milky, slightly acidic fluid containing citric acid. What is that needed for? More energy. ATP means making more energy. That is citric acid. It has many proteolytic enzymes like prostate, specific antigen, and acid phosphatase. What are those enzymes good for? It is to re-liquefy the coagulated semen, which happens 20 minutes after ejaculation. It also has an antibiotic for cleaning, and that is seminal plasmin. Why the hassle of arresting and then setting free? What am I talking about? I'm talking about the clot, and then what does to re-liquefy mean? The specificity is holding you, right? God is mentioning for specificity. Specificity, specificity, specificity. So, what does to re-liquefy mean? To make it more liquid, right? To bring it back. So, we arrest it. Once ejaculation happens, we have to clot. We stop those sperms from moving, and this is happening by the protein seminogillin, as you can see, coming from seminal vesicle. And then if you wait 20 minutes later, they become more moving and more liquid. Why the hassle of arresting and then setting free? Not to use up all the energy, right? Because you may be so excited that sperm rise, so you're excited to reach, of course, the egg for fertilization to happen. Then we lose all that energy in 2 seconds, right? So we have to arrest them, make them sit for a minute, and conserve the energy, and then you can set those sperms free again 20 minutes after that by using enzymes such as prostate, specific antigen, and acid phosphatase. And then we have something for cleaning, and that is the antibiotic seminal plasmin. My last stop is looking at bulbourethra or pulvers. It makes up to only 15%, which is very minimal. This is still alkaline that can protect the passing sperm by neutralizing the acidity. Urine is very acidic, so I have to buffer it and make it more friendly for sperms. In the lining of urethra, and then it has also a mucus, good for lubrication, the end of venous, and the lining of, are we good? Urethra. So, in conclusion, most of semen is acidic, or most of semen is alkaline basic. Acidic or alkaline? How come? 60% alkaline, 15%, which means 75, right? Simple math. 60 plus 15, 75% alkaline. Only 25% is acidic, if any, right? So the more dominant, of course, pH is alkaline because most of the secretions are alkaline. Only 25%, as you can see from citric acid, from prostate.